Hello and welcome to my presentation. My name is Syed Farhan Ahmad. I'm the lecturer at the Faculty of Science, Kesetsat University in Thailand. And today I would like to talk about the genomic applications in exploring the evolutionary dynamics and genomic elements in vertebrates. Genomics allows us to study and address the new questions of the genomic elements that are involved in the local adaptation, health, behavior, genetic differentiation of different vertebrate species. As we know that we are entering in population genomics era and the chromosome level genome assembly, which are near assemblies, and most of those are achieved by the vertebrate genome projects that would be provide a valuable bioresource for the bioresearch. So in our research, we are exploring the genomes of different models and non-model organisms to address different evolutionary aspects about the genomes of these vertebrates, including the chromosome biology, the organization of repetitive elements, genome-wide SNPs, 6 determination systems, and the development of microsatellites for understanding the diversity and population genetics. Today in this presentation, I would like to present and highlight about 2 cases of our snake genome and the macaques population genomics. And several of our research questions which involve the variable patterns of evolutions across different species and populations in vertebrates, and understanding their novelties and adaptations, how repeats can change the dynamics in the chromosome evolution, and what different systems evolve different sex determination. So first of all, the snake genome. Why is it important to study a snake genome? Well, because they are interesting and exciting models for studying the evolutionary features, and not only about their importance and biomedical importance in the snake bites most of the developing countries, but also in terms of the chromosome evolution and dynamics. Previously, we have highlighted that the chromosomal variability in reptiles are very unique, and they have a distinct karyotypic and genotypic variability in this, particularly snakes, which allow us to study the structural, functional, and evolutionary of chromosomal rearrangements. We map different Beck's clones from chicken genome on the snake genome to study the conserved homology, and also to study the chromosomal specific repeats, including the sex chromosomes. Currently, we are working on the genome assemblies with the long reads and short reads, spec bio and Illumina assembly. And we are using these reference genomes to understand the interspecies homologies of any large genomic segments and the mechanisms that would mediate the chromosomal rearrangement in snakes genomes, and how these events can impact the sex chromosome evolution. So we have achieved the high quality chromosome assembly of the Siamese cobra, which is an important species from the Thailand with an N50 of 222.6 MB. And we are annotating these genomes and providing as a reference for understanding the chromosomal homologies with several patterns of fusion and fissions, which can evolve as a result of can be established in the macro and micro chromosomes evolution, and also the 6 chromosomes with inversion-like patterns. And we propose to hypothesize that how the chromosomal rearrangements are mediated by different elements, including transposable elements and satellite DNA, leading to the diversity in species across different amniotic lineages. And another study, we are exploring the population genomics of wild macaques. We have collected high-scale population genomics data from multiple samples across different 380 individuals from different regions of Thailand, and which is a hybridization zone for the different cross-species hybridization. And we have genotyped using DD-rate sequencing data with enough coverage to study the genetic structure, diversity, and admixture, and to understand the population demographic and gene flow. With different population level analysis, including the PCA and the structure analysis, we found that high-level genomic admixture and long-tail macaques and the rhesus and long-tail macaques can also share the ancestral gene flow indicative of cross-species hybridization. And We also use different analysis such as FST and XPNSL to identify the loci that are under highly positive selection. And when we perform the gene ontology enrichment, we found interesting functions related to the brain development that could possibly suggest the brain evolution in these species. In another analysis of the demographic modeling, we found different trajectories in the different species, and we found that long-term macaque particularly experienced a recent decrease in the population size, showing the recent bottlenecks. And we also found that the gene flow between two subspecies, which indicate the hybridization and integration between the central as well as can be central force in the evolution of Macaca fascicularis aurea, which is a long-tailed macaque species. And with this, we emphasize overall taken together, these findings suggest that the genomic approaches can be applied, whether it is the population genomics, comparative genomics, chromosomes assemblies for understanding the interesting aspects of the evolutionary biology in vertebrates. Finally, I would like to thank for and acknowledge the PI and the whole team for the Kansas University, the AGB Research Center, and the funding agency. And also, I would like to thank the organizer of Biodiversity Genomics for giving me this opportunity to present our data and share our recent findings. Thank you.